biological functions, we can also assign their ages according to their member proteins. And just want to show you, from the function point of view, different ages, actually, they are quite separated to different functions. And I think this is also very follow our expectation, because for more and more complex organisms, when we say the complex organism like human, more of the operational definition of complex mean usually what we do is we just try to calculate how many different cell types were in this organism. For human, that human is the known organism with the largest number of different cell types. So that's why we always say that human is the most complex organisms. So there are many new functions like the not so complex, simple organism they don't need. So they don't need that kind of function. So all those functions, all those responsible genes will be older genes. But the youngest genes will be responsible for some new functions. And the last result and last plot is for each protein. Actually, not every protein are equally distributed inside a cell. So inside a cell there are some hierarchical organizations. So some of the protein will be expressed in the nucleus, and some of the protein will be in the cytosol, and some of them in the cell membrane, or some of them in some cell organs, cellular organs like mitochondria or others organelle. So from this, according to their cellular localization, you can see that. So the color is the gray one means the oldest one. So G1, G2, G3, G4, G6. So you can see that for nucleus, there's a large proportion of all these proteins, and also from the mitochondria, because the mitochondria was known to be a very ancient bacteria, was absorbed by eukaryotes. So very many old proteins. And then for the membrane, there are much smaller proportion of all these proteins, but more young protein. And also there are some proteins, once it's produced, it will be secreted to the outside the cell, because in our body, between the cell, actually there are many, there are also some structures. We usually call the extracellular matrix or something to hold the tissue structure. So many of them, the extracellular protein, they are younger proteins. So this, from the age point of view, so in the protein-protein interaction network, all these ones look like they are sitting inside the center part of the protein interaction network, and the younger one is in the outer area, periphery. And their location inside the cell actually also follow this kind of behavior. So I think this is all what I want to say today. So we just developed a method to try to, it's very simple, right? I think. We just got an idea, why don't we use the age to separate our protein interaction network? And somehow just nobody has ever done this. The previous, actually in 10 years ago, there's just one paper did this in the yeast protein interaction network, but much simpler. Then the thing I want to say is, I already said that all these ones are in the center of the network, and the youngest one on the more peripheral, and the same ages or similar ages protein, they like to interact with each other, so like social network.
So those are simulated to use the inverse square law.
Sorry?
All these all the interaction are simulated based on 1/r and the inverse square law.
That's just for visualization, just for visualization. Yeah. For all the analysis, actually, this protein-protein interaction network, don't think of it as a physical network. This is just an information network. They are not. Just visualization. Just for visualization, we need to input some physics. So most of the work are just done by my previous PhD student and some other master student and someone and my wife. They are our co-advised students. Okay, thank you.
Great. Questions?
So where do you see physicists can contribute this game? Like where else other than inverse Gaussian law and like the screen?
That's similar.
Simulation other than that? But, is there some actual idea of physics?
Actually, there are already many physicists study the protein-protein interaction network. You just try to build some model to explain why they will evolve to this kind of structure. And you can think of any crazy ideas. Because that's just the information network, not the real physical network. So you can think of some physical model, then to fulfill this physical model, their result will turn out to be this kind of information. There are already quite a few papers more than 10 years ago, some physicists are doing this. At that time, it's a very hot topic. So quite a few physical regulator papers.
So what's the hot topic now?
For physicists?
Yeah, basically in this game, in this line of like this. Is it trying to answer the connection between different species, like as what you were studying? That's considered a very
Currently,
the main thing.
the one hot topic for theorists to do biological problems, one of them is not really on this network, but for example, how cancer cells evolve. Because the cancer development is like evolution, a very small time scale evolution between all these cells. And all these cancer cells actually they evolve very, very fast. So I know many mathematicians, theoretical physicists, they jump to this game because just in these 2 or 3 years, there are new technology allow us to do the single cell sequencing. So you can have like thousands of cells, all that sequence, extract from the real cancer patient tissue. Then you know all these, how their sequence changes.
So
then you can try to build the model.
Generations.
Yeah,
Other generations.
No, you just like what evolution people do from, because right now we only know many different organisms, right? We don't see their history.
Yeah.
So we need to compare them to how to say, to transcribe, to predict the history. For cancer the same. You just from the same tissue, different condition, they will have a different distribution of different types of cancer cells, different genome sequences.
Do you still see the causal or like a scale-free kind of behavior in this kind of?
I myself
Short
study that.
Short time.
I myself usually more focus, try to think like a biologist, try to simulate. I don't really dig into all these, how to say, the real physics interest problems. Because actually, even for the topic I talked today, because I'm coming here to give a talk, I just select the one which contains the least biology and simple. But I think most of the biologists I think no one likes this kind of study. They just think that's this has no application capability. They don't know why I like to know this.
Other questions?
Why? So this is very relevant in the real. I mean, all this coding technique shouldn't that be applied to, for example, like a word? Okay, I mean more complicated context, but like a word recognition, that many different words have the same meaning, like
Right.
in a different group in different ways, and the reason for saying that is, I mean, if once you can train a machine to do that, you can
That's the current most hottest topic.
Well, yes, basically.
Yeah, because everybody know AlphaGo, right?
Yeah.
Because AlphaGo depends on the current hot topic called deep learning. So just starting last year, already there are a few Nature papers publishing using this kind of deep learning techniques on this genomic sequencing.
That just sounds like natural language processing.
Yeah. Natural language processing also how to say a hot topic for deep learning application.
And if you want to make a diagram, you can do it offline.
Yeah.
Any further
Sorry.
questions? I do have one. Facebook is not the full world. So your PPI is also an oversimplification.
Right.
Now the network, you take a view of it, but there's much more communication going on. So how would that reflect in this kind of analysis?
Yeah, the one thing is actually even for this information network, it's also already not complete because many protein-protein interactions are not measured yet. So this is indeed oversimplified. However, it has already been some studies showing that even you try to like randomly remove a large portion of this, all this, we also did this. Always when we publish a paper, we need to do this kind of validation. All these observed results doesn't depend on even I remove like 30% of the data, I still observe the same thing. So I think this should be just for the result. I sure should be solid enough. However, this is just like a simplified overview of the global system. So that cannot really, for example, if I really want to say, oh, indeed, actually, in which disease, which part of this network is really important, I can only say, oh, probably the center part, right? However, really want to answer those, what we usually do, we need to combine other type of biological data. Because for all specific problems, PPI is just one of the information. So this is kind of framework, just like Google Map. Google Map is just all these rows, just a framework. However, for each time, for which row there are how many cars on this, what's the flow rate, and there are other measurements we can utilize. Then just combine those with this, then we can dig out all those more detailed information.
Okay. Only student questions? Usually there aren't, but gotcha. So we had a different snapshot of different life forms. So let's thank Professor Huang again.